Good day, everyone. Today, we are going to discuss the microscope with emphasis on its principle, parts, use, and care. Let's start by answering the pre-lab discussion.
Now we have the microscope parts. First is the ocular lenses. It is a cylinder containing two or more lenses. It helps to bring the object into focus. Second, diopter adjustment. It is used to correct the differences in a vision an individual may have between their left and right eyes. Third, nose piece. It holds the object lens in place to rotate and can be changed easily. Fourth, objective lenses. It magnifies the object 4 times, 10 times, 40 times magnification. Fifth, slide clip. Holds the slide in place on the stage, controlled by turning two knobs. 6. Coarse focus knob. It is used for initial focus on the object, low power adjustment, and then adjusting the stage. 7. Fine focus knob. It is used for final and clear focus on the object, used for delicate, high power adjustment that will require fine tuning.
So for the next part of microscope, we have aperture. This is located under the stage as the light travels from the illuminator. It passes through the condenser which is focused on the specimen. This is also the measure of its ability to gather light and to resolve fine specimen detail while working at a fixed object or specimen distance. Next is light source. This is usually situated in the base. The brightness can be adjusted. Next part is iris diaphragm. It controls the amount of light going through the object. This is an opening and closing adjusts the diameter of the light beam. Next is mechanical stage. It is where the glass slide is placed. It can be moved upwards and downwards. It also has a hole to let the light to pass through. Next part is head, the upper part of the microscope. It houses the eyepiece and objective lenses. Next is arm. This is a rigid structure for stability. It is used to carry the microscope. Next is the base. It supports the microscope and houses the light source. Also, lower part of the microscope.
The part of a microscope primarily responsible for magnifying objects or specimen is a combination of lenses found in the objective lens system. These lenses are located near the specimen and are designed to gather light and magnify the image of the specimen. Microscopes often have multiple objective lenses with varying magnification powers, allowing users to select the level of magnification they require for their observations. The eyepiece lens or ocular lens also contributes to the overall magnification by further enlarging the image produced by the objective lenses.
Resolution refers to the level of detail contained in an image. It also refers to the capacity of a microscope to identify two objects that are narrowly spaced apart as distinct entities. While magnification is important to see what we're looking at under the microscope in an enlarged manner, it would be hard to accurately observe and analyze the specimen if it appears blurry and lacking in detail since the resolution is poor. Therefore, resolution is more crucial than magnification for obtaining relevant observations in microscopy since it guarantees that tiny features are seen clearly and properly.
So, an illuminator, also known as a light source, is a vibrating mirror that reflects light from the sun or other external sources. And the most commonly used mirror have flat and concave sides. This particular type of mirror is useful because it focuses the light rays and magnifies the image. They reflect light that illuminates them, producing a magnified and sharp picture of the specimen.
On this laboratory activity, our professor gave us a different specimen that has been observed under the microscope in different magnification. The scanning or viewing, low power, high power, and the oil immersion. There are four types of specimen: the bacteriology, parasitology, botany, and histology. The first one is bacteriology. The first specimen in bacteriology is Vibrio. Vibrio bacteria are natural part of marine and estuarine environment. They are commonly found in coastal waters, particularly in warm waters. Vibrio species can also be found in brackish water, such as estuaries, and in seafood, especially oysters. The bacteria can enter the body through the consumption of contaminated food or water, which can cause poisoning, leading to infections such as cholera or wound infections. The second slide is the Corynebacterium diphtheria. Bacterium that can cause diphtheria, a serious respiratory illness. The bacterium produces a toxin that can cause a thick, gray coating to form in the throat and nose, making it difficult to breathe and swallow. Diphtheria can also lead a complication affecting the heart and the nervous system. Vaccination against diphtheria is highly effective in preventing the disease. The third slide of specimen is Bacillus subtilis smear. Sample of bacterium Bacillus subtilis that has been spread thinly and evenly on a slide or other surface for microscopic examination. Bacillus subtilis is a common and beneficial bacterium found in soil and gastrointestinal tract of human and animals. It is often used as model organism in scientific research due to its genetic and physiological characteristics. Smear of Bacillus subtilis may be used in laboratory settings for studying bacterial morphology, motility, and other characteristics. The next slide of specimen is Mycobacterium tuberculosis. Mycobacterium tuberculosis is a bacterium that causes tuberculosis or TB, a contagious airborne infection that usually affects the lungs but can also affect the other parts of the body. TB spreads through the air when a person with TB of the lungs or throat coughs, sneezes, or talks, and the other breathe in the bacteria. TB can be latent, where the bacteria are present but inactive and not causing symptoms, or active, where the bacteria are multiplying and causing symptoms. Active tuberculosis can also be treated with antibiotics, but it requires a long course of medication to cure. The next specimen is Diplococcus pneumoniae. Diplococcus pneumoniae is also known as Streptococcus pneumoniae, is a bacterium responsible for many types of pneumococcal infections. These bacteria are typically found in the upper respiratory tract of humans, but they can cause serious infections such as pneumonia, meningitis, bacteremia, the bloodstream infection. Pneumococcal infections can be prevented through the vaccination. The last specimen in the bacteriology is Streptococcus pyogenes. Streptococcus pyogenes, also known as a group of Streptococcus, is a bacterium that can cause range of infections in humans. This infection can vary from mild, such as a strep throat or impetigo, to more severe and life-threatening, such as necrotizing fasciitis, the flesh-eating disease, a streptococcal toxic shock syndrome. Treatment typically involves antibiotic, and in some cases, surgery may be necessary to remove the infected tissue. Now let's go to the next part, which is the parasitology. The first specimen in parasitology is Balantidium coli tropozoite. Balantidium coli is a large xylated protozoan parasite that can infect the digestive tract of the human and other animals, causing a condition known as balantiasis. The tropozoite stage B coli is the active feeding form of the parasite that lives in the colon of the host. Infections are usually asymptomatic but can cause symptoms such as diarrhea, abdominal pain, and in severe cases, dysentery. Treatment typically involves the use of antibiotics. The second specimen is monocytes. Monocytes is a genus protozoan parasite that primarily infect earthworm. These parasites are characterized by having a single large cyst-like structure in their life cycle. Hence the names monocytes. They are typically found in the reproductive organ of the earthworm, where they can cause infection known as monocytidiosis. Monocytes organisms are not known to infect human. The third specimen is Trichinella spiralis larva. Trichinella spiralis is a parasitic roundworm that causes trichinelliosis, also known as trichinosis, in human. The larvae of Trichinella spiralis are the infective stage of the parasite. When infected meat, typically pork, is consumed, the larvae are released in the intestine where they mature into adult worms. The female worms produce larvae that can migrate to the various muscles where they form cysts. Symptoms of trichinosis can include gastrointestinal issue, muscle pain, fever, and swelling around the eyes. The fourth slide is Ascaris male. Ascaris lumbricoides is a species of parasitic roundworms that infects humans. Ascaris worms are dioxous, meaning they have a separate male and female individuals. The male Ascaris lumbricoides worms is smaller than the female and has curled posterior end, which used to grasp the female during the copulation. Male Ascaris worm have a curved tail, whereas female have a straighter and thicker tail. The second to the last is Himonolepis diminuta gravid, or what we call is Himonolepis nana, also known as the dwarf tapeworm, is a species of tapeworm that can infect humans. The gravid stage refers where the tapeworm is pregnant, meaning it is filled with egg. The next specimen, or the last specimen in parasitology, is Schistosoma japonica WM. It is a species of parasitic flatworm that causes schistosomiasis in humans. The term ova refers to the egg of the parasite. WM stands for the whole mount, indicating that the specimen has been prepared for microscopic examination by mounting it intact on a slide. Therefore, this refers to a slide containing a whole mount specimens of the eggs of Schistosoma japonicum for the microscopic study.
Next, we have is botany. Botany is the scientific study of plants, including their physiology, structures, genetics, ecology, distribution, classifications, and economic importance. So first, in the botany, we have the alimceparoot tip longitudinal section. You would see various cells in different stages of cell division like mitosis if you observe an onion root tip longitudinal section under a microscope. The cells will have a rectangular shape with visible cell wall, cytoplasm, and a large nucleus with a dark staining nucleolus. Next, we have the onion skin plasmolysed whole mount. A plasmolysed onion skin cell will exhibit a shrunken cytoplasm pulled away from a cell wall due to osmosis. The cell wall will be visible as a distinct outline surrounding the contracted cytoplasm. The nucleus may still have visible, but other organelles will be difficult to distinguish. Third is the penicillium. Penicillium consists of branching filamentous hyphae. These hyphae are made up of cells that have a cell wall but lack many of the organelles found in the animal cell. You might also see spores, which are reproductive structures produced by the mold. Next is the helianthus stem cross-section. A sunflower stem cross-sections reveal various tissues working together to support the plant and transport materials. You would see the epidermis, the outermost layer of the cell, which is covered by a waxy layer called the cuticle. Beneath the epidermis is the cortex, which is a layer of parenchyma cell that provides support and storage. Vascular bundles, which transport waters and nutrients, would be visible throughout the cortex. Each vascular bundle contains of xylem, which transport water, and phloem, which transport sugar. Next is the spirulina, whole mount. Spirulina is a filamentous multicellular cyanobacteria. Under a microscope, you would see a trichome, which is a chain cylindrical cell. The cell contains chlorophyll, which gives spirulina its green color. These cells also contain a variety of other pigment and nutrients. Next, we have the histology. Histology is the microscopic study of tissues and organs through sectioning, staining, and examining those sections under a microscope. Often called microscopic anatomy and histochemistry, histology allows for visualization of tissue structure and characteristic changes the tissue may have undergone. First, in the histology, we have the mammal pyloric stomach section. A section of pyloric stomach would show a stratified epithelium lining the surface. The epithelium would consist of various cells, including mucous cells that secrete mucus to protect the stomach lining, and chief cells that produce epsin, an enzyme that helps break down protein. Muscular layers would be visible beneath the epithelium. This muscle contract to churn food and help move it into small intestine. Next, we have the small intestine cross-section. The lining of the small intestine would be visible under a microscope. This lining is thrown into fold and finger-like projection called the villi, which increase the surface area for absorption of nutrients. The epithelium lining the villi would be visible, and you might also see goblet cells that secretes mucus and enterocytes that absorb nutrients. Next, we have the mammal spinal cord cross-section. The mammal spinal cord cross-section would reveal gray matter in the center and white matter surrounding it. The gray matter consists nerve cell, body, and dendrites, while the white matter consists myelinated nerve fibers. The spinal cord would also be surrounded by a tough membrane called the meninges. Next, we have the mammal fundic stomach section. The fundic stomach epithelium consists of columnar epithelial cells. These cells include mucous cells that secrete mucus to protect the stomach lining, and parietal cells that produce hydrochloric acid, which help to break down food and kill bacteria. Blood vessels would also be visible within the tissue. Next, we have the frog stomach section. The frog stomach lining is similar to the mammalian stomach lining in that it consists of epithelium cells that secrete mucus and digestive enzymes. However, the frog stomach epithelium is simpler in structure and does not have as many different types of cells. Lastly, we have the artery medium cross-section. The artery cross-sections reveal three distinct layers: the tunica intima, the tunica media, and the tunica externa. The tunica intima is the innermost layer and consists of a single layer of endothelial cell. The tunica media is the thickest layer and is made up of smooth muscle cells and elastic fibers. The tunica externa is the outermost layer and consists of connective tissues.